Dear audience, today we are starting a series of lectures of the Department of Histology, Cytology, and Embryology of the Yaroslav State Medical University. The topic of our first lecture is the subject of histology as a fundamental science and the significance of this discipline for medical education. The first issue needs to be considered is what histology is as a fundamental science and as an academic discipline. Histology is a branch of biology dedicated to studying the regularities of development, structure, and life activities of tissues in multicellular organisms. Histology as an independent science emerged in the mid-19th century with its own. It has its object and its specific research methods. Histology has its scientific language, theories, laws, and concepts. Histology includes a number of sections: plant histology, animal histology, evolutionary histology, veterinary histology. For a medical university, the section of medical histology is of primary importance, a leading academic discipline focused on studying the human organism. The objects of study of histology are multicellular organisms, and the main subject of research in this scientific discipline is the concept of tissue. Tissue is a system of specialized cells and their derivatives that emerge in the process of evolution, united by the community of development, structure, and functions performed. Tissue is a private system of an organism corresponding to one of the levels of its structural organization. It fits the intermediate position in the pecking order: molecules, cells, tissues, organs. Tissue contains cells that are principal elements of tissues, as well as cell derivatives. Tissue has specific functions that are carried out through the joint activity of all its elements. Tissue is a cell system, but not a simple sum of cells. Tissues were formed and their properties were fixed during the evolution of multicellular organisms. In representatives of different evolutionary groups of animals, tissues specialized in performing the same basic functions acquired a similar structural organization. Various body tissues, more than 60 types, may be classified by structural properties and functional determination into four morphofunctional tissue groups. Epithelial tissues constitute cell strata, exert the barrier function, provide for external metabolism, are able for secretion. Connective and supporting tissues are marked by a well-developed intercellular substance. This tissue group provides internal metabolism and homeostasis, play a sustentacular role. Muscle tissues are constituted by muscle cells or muscle fibers. They are responsible for all types of motor activity in the body, the movement of the body and its parts in space. Nerve tissue consists of neurons and glial cells, ensures an integration and regulation of cell activity and organ functioning. The role of tissues in the body is significant. Firstly, tissues serve as the building material of organs. Within each organ, a variety of tissues can be found, shaping its unique structure. For instance, in a histological specimen of the tongue, we can observe the representation of all tissue groups I have previously mentioned. The organ's surface is covered with protective epithelium, while beneath it, a specialized glandular epithelium is present. Loose connective tissue is situated below the epithelium. The foundation of the tongue is composed of skeletal muscle tissue. Additionally, clusters of nerve cells can be found here, regulating gland secretion, muscle contractions, and the metabolism of the surrounding connective tissue. In certain cases, tissues can function as an independent system of the body, such as the blood system and the connective tissue system. Blood is a tissue that circulates throughout our entire body without a specific organ localization. Connective tissues are present in almost all organs and share common properties. In some pathological conditions, connective tissue damage can occur throughout the body. This is why diseases of the blood system and diseases of the connective tissues are distinguished. Cells are the main structural element of any tissue. It is the cells that determine the basic properties of tissues and give rise to non-cellular structures called cell derivatives. In any mature tissue, different types of cells naturally differ in shape, size, and the presence of specialized structures. For example, nerve tissue cells are characterized by numerous processes, while blood leukocytes contain specialized granules in their cytoplasm stained with various dyes. Muscle tissue is characterized by the presence of contractile structures called myofibrils. The epithelial cells of the respiratory tract contain numerous actively functioning cilia. Overall, the appearance of specialized structures during tissue development is denoted by the term cell differentiation and is the result of genetically programmed synthesis by mature cells of specific structural proteins and other substances. Cell differentiation occurs especially intensively in the embryonic period. Derivatives of cells are also a crucial element of tissues. Their diversity can be divided into three main groups. Firstly, there is the intercellular substance or matrix, which is the result of the synthetic activity of specialized cells. Primarily, this includes collagen and elastic fibers, as well as the amorphous substance of connective tissues filling the space between them. Postcellular structures are the result of terminal cell differentiation. Red blood cells and blood platelets do not contain nuclei and maintain viability for a certain period in peripheral blood. Other postcellular structures, such as the horny scales of the epidermis, hair and nails, are formed as a result of programmed death of mature epithelial cells. The third type of derived cells are syncytia. They are formed as a result of the fusion of numerous individual cells. In this case, the cell boundaries disappear and a common cytoplasm with a large number of nuclei is formed. A classic example of syncytia is the fibers of skeletal muscle tissue. After we have discussed the basic properties of tissues, let us focus on the methods of studying their structure. Due to the small size of tissue elements, it is practically impossible to study them in detail with the naked eye. So the main method of their investigation is microscopy. The most commonly used method is light microscopy, based on studying specimens in transmitted light. Light from the source passes through the specimen being studied, is directed into the objective lens. The magnified image is observed by the researcher through the eyepiece or displayed on a monitor screen. Maximum magnification of light microscope is 1,300 times. Resolution or minimal distance between two neighboring objects when both of them can be seen is not high, about 0.2 micrometers. To study samples of organs and tissues under a light microscope, they must undergo a special processing aimed at creating a high-quality histological specimens. A histological specimen is a slice of an organ with a thickness ranging from 5 to 10 micrometers after pre-processing and staining with specialized dyes. The main task of the researcher is to ensure that the obtained histological specimen closely resembles the in vivo structure of the organ being studied. Various algorithms for preparing biological samples for microscopy have been developed to achieve this goal. You will get acquainted with the main stages of the manufacture of histological specimens in detail during practical sessions. Since the light microscope allows effective magnification within 1,000 times, to more detailed study cells and tissues have developed electron microscopy techniques. Transmission electron microscopy is the main method based on examining ultrafine sections of specimens in E-beams emitted by an electron gun under high voltage and deep vacuum. Focusing is attained by means of electronic lenses. Image formation takes place on the photodetector. Images are saved digitally. Maximum magnifying power for tissue sections is in the range of 150,000 times. Resolving power is 1 to 2 nanometers. Electron microscopy of tissues usually uses magnifications ranging from 3,000 to 60,000 times, which allows to study the general appearance of cells and the subtle details of their structure. Scanning electron microscopy is a method based on scanning the surface of the object under study by E-beam. Secondary electrons dispersed or radiated by the surface of the object are registered by the detector to present a 3D image on the microscope display. Scanning electron microscopy of cells usually uses magnifications ranging from 2,000 to 30,000 times. This method is widely used to study the surface details of biological objects. For example, this is how connective tissue fibers and the outer surface of the cell nucleus look in a scanning electron microscope. At the level of light and electron microscopy, we work with thin, almost two-dimensional sections of three-dimensional objects. When studying a histological specimen, it is essential for the researcher to understand the actual three-dimensional structure of the microscopic object being studied. The lower illustration contains two microphotographs of the same small blood vessel, an arteriole. On the left is an image obtained through a transmission electron microscope, and on the right is a three-dimensional view of the same arteriole in a scanning electron microscope. The upper diagram shows variations in the sectioning of tubular structures depending on the plane of the cut. For example, different sections of the skin sweat gland can be visible as longitudinal cell strands, as well as transverse or tangential sections of epithelial tubules. The progress of biomedical and computer technologies in the 21st century has provided the technical capability to create high-quality digital copies of histological specimens using specialized robotic devices called histological scanners. The operation of such a scanner is demonstrated in the video. The system scans the entire histological specimen, and the individual images of the whole section at different magnifications are automatically stitched together into one large virtual slide. This digital visualization technique is called as whole slide imaging, WSI, and the navigation through digital samples is designated as virtual microscopy. Studying the virtual slide completely reproduces the process of light microscopy, but now it takes place on the computer screen. The active implementation of digital technologies in medical education is a priority direction for the Department of Histology, Cytology and Embryology of the Yaroslav State Medical University. In 2018, a project for full-scale implementation of virtual microscopy into the educational process as an alternative to classical light microscopy was first implemented by us in Russia. For this purpose, a collection of more than 300 virtual slides has been created and an electronic practicum, virtual histology for medical students, has been developed. Currently, our university is the only medical university in Russia where the study of histological specimens is fully transferred to the mode of virtual microscopy. Therefore, you have an excellent opportunity to gain a solid knowledge of histology through this most modern technology of visualizing microscopic objects. Along with working in the classrooms, you are given an opportunity to have open round-the-clock access to the digital resources of our department via the Internet. For this purpose, a specialized English language website, Virtual Histology for Medical Students, has been developed. After its loading, a student can open the electronic practicum and remotely work with virtual histological specimens. On the HistoNavigator channel, watch video lectures and educational films on working with histological specimens, as well as test their knowledge with the How Do You Know Histology training program. The algorithms for working with these resources will be shown on the next slide. For remote study of histological specimen collections, go to the slide box section of the electronic practicum, select the course section, and activate the corresponding hyperlink, Step 1. Step 2. After the page loads, choose the name of the histological structure from the list in Window 1 and activate hyperlink. In Window 2, a textual description of the slide will appear along with a hyperlink to download its image. Step 3. After activating the virtual slide hyperlink in Window 2, a digital image of the microscopic structure will be loaded into Window 3. Carefully read the description of the histological specimen and while navigating the virtual slide, locate the structures according to the task. On the HistoNavigator channel in the Histology in English playlist, you can access current histology lectures at any time, listen to them multiple times if necessary at an optimal playback speed. Additionally, the playlist includes educational videos on working with virtual histological specimens. How Do You Know Histology is the first Russian educational and training program on histology for the Internet. How to use the program. Step 1. On the main page, select the type of tissue to test your knowledge and activate the hyperlink. Step 2. On the next page, choose the task you need and activate the corresponding button. Step 3. After loading the task, carefully study the histological structure in the left window and then choose the best answer from all the options in the right window in each section. Information about the correctness of your choice will appear in a separate window. The course of medical histology will consist of 4 stages. The first stage involves revisiting and deepening knowledge in cytology or cell biology. The goal of this stage is to acquire solid knowledge of the principles of organization and the characteristics of the structure and functions of cells within tissues. The second stage entails studying general embryology as an introduction to general histology. The aim of this stage is to gain knowledge of the mechanisms of embryonic histogenesis, leading to the formation of the entire diversity of cells and tissues of the future organism from a single zygote. After studying cytology and general embryology, we move on to general histology, which examines the regularities of development, the structure and functions of the main tissues of our body. Having studied the properties of tissues, we will move on to the largest section of the course, special histology. By studying the microscopic structure of organs, you should get a comprehensive knowledge of the structure and development of human organ systems within the normal range. The systemic significance of histology in higher medical education is immense. Firstly, histology provides knowledge of the structural foundations of cell and tissue organization, essential for the study of other fundamental medical disciplines, such as normal physiology, biochemistry, immunology, and pharmacology. Fundamental knowledge of tissue and organ structure in the norm is necessary for understanding the pathogenesis and morphogenesis of diseases. In this applied sense, histology serves as an introduction to pathology. Equally important is the role of histology in the advancement of biomedicine, as the achievements of modern medical science are largely based on progress in studying properties of cells and tissues. Histological and psychological methods are widely used in clinical diagnostics. Assisted reproductive technologies based on working with germ cells and embryo cultivation are actively developing worldwide. The synthesis of biologically active substances in cell cultures is the foundation of modern bioindustry. The development of new pharmaceuticals based on the study of cell metabolism and intercellular interactions is a key direction in the development of modern pharmacology. The achievements of histology open up broad prospects for the development of future medical technologies. Cellular tissue engineering aims to create artificial implanted organs, while regenerative medicine is developing methods for restoring damaged human tissues and organs. Dear students, successful mastery of the histology course will require significant efforts from you to acquire new knowledge about the subtle details of the human body structure. In the process of studying the achievements of this fundamental science with the help of new digital technologies, a new fascinating world of cells and tissues will be revealed before you. Welcome to Modern Histology. Stay with us for our next meetings on the HistoNavigator channel.